we grow them, and we study, or we try to express the enzyme, express the antibiotics, and we can use that, right? But the problem is that when we take the microbes from the soil, for example, from the soil, if we try to put on the agar, the problem is that only less than 1% can grow. So, for example, we have 1,000 types of microbes in this soil. If we try to grow on the agar, perhaps only 10 or 20 microbes can grow. The remaining of the 99% cannot grow because of the media, the nature, the pollution. These microbes cannot grow. So we only do whatever we have, the potential, the enzyme, the drugs, everything that we have at the moment is only on the based on this 1% microbes. So imagine if we can tap the remaining of the 99% of this microbe agriculture, so the potential will be very huge. We can do a lot of things with this potential, like the remaining 99% of what we call is the non-functional microbes. Okay. Right. So the question is how we look into this 99%. Right. So this is what we call as metagenomic studies. Okay. So metagenomics has been around a very long time. Initially, they started with using traditional method like cloning and expressing the material. So basically, what is the metagenomic? Metagenomic is by definition is a genetic study. You study the organism where you have the genetic material, in this case the DNA, recovered directly from environmental samples, meaning that you don't touch it. So, example, you take the soil, you just straight away put on the extraction medium, you extract the outcome of the DNA. You don't culture it, you skip the culture because the culture you only get 1%. So, what you did, you don't culture it, extract all the DNA. So, the DNA will be a mixture of microbes. Okay? And then after that, using this DNA, because it's a DNA metagenome, you can study that. So traditionally, you clone it into a special vector, cloning to other vector and try to expression, sequencing, and so on. Okay? But nowadays, in year 2000, we have an NGS sequencing platform where you can make things even simpler. You don't have to clone it. So from the DNA metagenome, in this case, for example, so from this, the mixed microbial communities like soil, water, air, right? So we can see there's a lot of microbes, green microbes, yellow, blue, red microbes. You straight away do the DNA extraction, and from here we'll get the DNA of this bacteria. Okay. And next, what you can do is using the next generation sequencing platform, you can either use an empirical sequence analysis or metagenomic sequencing. So both using the sequencing approach, just that they have a different strategy on how to sequence this sample. Okay. But you can see here, we don't do any culturing steps. We straight away from sample to extraction to sequencing. There's no culturing process. So this is what metagenomic is about. Okay. You don't see culturing anything. Right. So the question, when everything is mixed, how are we going to start that? So that is why we have do this we call it because sequences or metagenomic sequencing. So how does this work? I will have a later explanation on the later, but briefly, what does this empirical sequencing about? So for empirical sequencing, we just sequence part of the DNA sequence. We call it as a DNA marker. Right, there's a specific DNA marker which are present across all organisms. For example, I give you an example. How to differentiate between a human and a plant? Human, we got head. For example, we have head. So meaning that this is the like which is a head, we call it as a human. So the plant doesn't have head. So we have like kind of a signature to call it as the organism. Same goes for bacteria. So bacteria, we look at the signature. So there's a DNA signature. For bacteria, we have an example CPS. Then we call it for bacteria. For fungi, they don't have a CPS. They have IPS. Then we give the IPS as signature to other functions. Right. So we call it as a DNA marker to identify this organism. Okay, so because you just look at the small DNA marker, right? So the cost is very low, cheap, right? We can do it to the microbiome taxonomy and to look into the abundance, how many they are in the environment, right? However, because we only sequence just a small part of the DNA, we don't have which we can define the taxonomy, the identification. We don't have other information because we only target the information. So we don't have functional information. What is functional information? It's like the gene, what's the enzyme, so we left out the information. But good enough for you to do taxonomy identification. It's cheap, fast, but only taxonomy identification. The other one will be the metagenomic sequencing. So this will be very straightforward. So from the DNA extraction, so using the NGS sequencing platform, we just sequence the code. And using the biogenetic analysis, we try to patch up everything, try to reconstruct back the genome. And from there, we can do the taxonomy identification as well as the functional information because we sequence everything. But because we sequence at a very high amount of data, so the cost is very expensive. So that's a trade between these two approaches. So it depends on the objective, what are you looking for. If you're just looking for the taxonomy, what's the bacteria inside the environment, you don't care about the gene information, you can go for a sequence analysis. But if you need both the taxonomy and also the enzyme, the gene information, then you can go with the metagenomic sequencing. So that's a trademark between these two approaches. Okay, right. So just a brief comparison of these two approaches. As I just to recap some of the part. So amplified sequence analysis and shotgun metagenomics. What's the difference between these two? So amplified sequence is cheap because it's low in coverage. Shotgun metagenomics is expensive because you sequence more. And then for amplified sequence analysis, the genomic identification limit to the gene target. Right? So only limit to gene target. You don't have functional information, no gene information. Right? And then for shotgun metagenomics, you don't have any limit on identification because you sequence everything. Inside the data, you will get fungus, fungi, bacteria, eukaryote, viral is there, so you will get the information. So there's no limitation. And for function information, you will have the functional information because you sequence everything. Right? Yeah, that's some of the highlight comparison. There are a few more. Okay, but just for knowledge purposes, I will bring up those most important. Okay, right. So now we look into the application. What we can do on this metagenomic study. Okay, one thing what we can do is we can do for environmental monitoring. Okay, so what does it mean by environmental monitoring? So, for example, let's say you have a farmers, right? They plant paddy. The problem is that one plot have a very bad yield, another plot have very good yield. So there are few factors that contribute to this. One of them, it could be because of the microbes in the soil. Could be something's missing. Maybe the soil, they put too much fertilizer and then they kill all the microbes inside. Right? But how do you know what the microbes inside there is missing? Which one is which one is missing? So we can do metagenomic study to look into that. Right? So what we can do is we can take soil on the bad yield crop and we good yield. We look into we send for amplified sequence analysis, right? And we got the species information, the abundance information. We can compare them both. So whatever is missing in the poor microbe, poor soil, we assume that it could be the contribute to the bad yield production. That's one example. Another example will be the pathogen detection and diagnosis. So you have like this tomato, for example, you can see there's an infection, but you don't know what is that. So what you can do is cut this part and you send for amplified sequence analysis. Since it could be a fungi, you can use IPS, and from the result will show you what's the topmost of the fungi in this area. So you assume that this could be caused by the infection. By knowing what's the fungus, what's the cause of this infection, we can give you a proper treatment, the correct treatment plan. All right. So that's another example. Another example will be in the industry, like in the like a fermentation. So fermentation, you put microbes, what do you call this? beneficial microbes in your food to make the ferment food. Even like the beer production, so we use some yeast, right? So we want to make sure that the quality are remained in the process throughout the process from one batch to another. So we want to make sure there are correct yeast. So you can also use the metagenomics studies to look into that. Okay. Also for the university research institute, for any fundamental study, anything that comes from a mixed sample. Let's say in your lab, you do some culturing, you have your bacteria culture, right? But one day you found that there's a mix of microbes inside there. So you want to know what's inside this mixed microbes. You can do an ampli-consequence analysis, and they will tell you what other species are inside the samples. Okay. All right. So this is just an example of the metagenomic application. There are a few more. Feel free to look into the paper. You can type metagenomics and you can see a lot of papers about. All right. Okay, so now let's look into the ampli-consequence analysis, focusing on the ampli-consequence analysis. All right. So this is the workflow. Actually, the workflow is very straightforward. Okay. Again, so there's basically our split into three parts of the process. So the first process is sample preparation. It's very important. And I think that we do in the lab to start with a good sample preparation. Bad sample preparation and good bad results. So we need to make sure that sample preparation are well, so that everything will be nice and good. Okay. So from the for the sample preparation, so we do a direct genomic isolation, direct genomic extraction from the environmental samples. So in this case, the soil, the water, the air, right? So we do a direct DNA extraction. Okay, so we have the soil, and then we extract the DNA, the outcome will be the DNA of the mixed sample, because DNA metagenome. The DNA metagenome quality should be good enough and high enough for you to do the PCR. Okay, right. So normally for PCR, we don't need that much, as long that you have able to PCR the sample. Normally like 200 nanogram will be as a good indicator that this precisely good. Okay, and the OA264 280 is for 1.8.0. Okay, for first phase, if you would like to do this service to us, so we require the customer to send this amount of data. But if you have any difficulties on extracting the DNA, we do have service for the DNA extraction. Right? So we do accept a lot of type of samples, like animal tissue, cell colors, soil. So if you have any inquiry about sending the tissue sample, you can talk to Nam Anh. So they will advise you on how to send the sample. Right. So now we already extract the DNA. The next step, what you can do is you need to do the PCR. So you have the PCR, the DNA marker. I mentioned just now, the DNA marker is very important here. Right? It's very short and they are very precise. Okay. And right, talking about the PCR. Okay, so the PCR, this is a very the features are short so that it can be sequenced in the next gen platform. They are present across the group. Okay, so for example, the bacteria we have CPS. All bacteria has CPS. For fungi, they all have IPS. Algae use APS. Animal, you use COX1, Saccharomyces oxidase. And for plant, you can use the RBCL or McKay. So these are the DNA markers that we can use to target the organism. Right. So, for example, we have a soil. You want to study the diversity of bacteria and fungi in the same soil. You can use two sets of data for the DNA metagenome. You can PCR the CPS, right? And the same sample, you can also sequence the IPS. So, with this data, we can have information on the bacteria and also the fungi together. Okay. Right. So, yeah, we have to do the PCR amplification. This is specific of a DNA marker. Right. So now we have the PCR product. Okay, so it's fine. So we have the PCR product. The next thing what we should do is we need to sequence this. And for sequencing to be done, we need to do the library construction. Right, like getting the adapters and put it in the sequencing machine. So we can sequence on Illumina platform, PacBio, none of all depends on the objective and also the price as well. Okay. Okay, so that's the stage 2. Right. So at the end of the stage 2, the biometric part. So at the end of the sequencing process, what you get is the sequence data. The sequence, the APDC in a FAST2 or FAST8 format. Right. Right. So the imaging is the sequence data. So this sequence represents of the what you have inside your sample. Okay. So imagine that the blue color is the DNA, the sequence from the blue color. This is the green one, came from the green, genome. The orange came from the yellow one, and also red from the orange genome. And it depends also how many of these bacteria present in the sample. The more you have, let's say, the more of the green color present in the sample, the more you will get here. Right? So it will be relatively present. Okay? But at the end of the stage, when you get the sequence, you can't see it yet, right? So we have to process it in the analysis part. Okay. In the stage 3, what we do is we use the computer tools to analyze this data, the sequence. So briefly, what the analysis do is they will cluster the sequence, taking as the representative of each of the sequence present, and they will count how many of them present in the sample. Okay, and since that you have the sequence, we can match it to the database like a CBI or C1 database, which contain the information, the taxonomy information, species information, bacteria information, fungi information. So you combine all this together, what you get is the sequence, the abundance, how many they are in the environment, as well as the species information in one table. So we have the relay out all the information. So we have, for example, we have to mix contaminated samples. They will let you know. The top one will be this microbe and the name of species. The second one will be this microbe and the species, and so on. So we have one. I will show you our demo report so you can see how the result looks like later. Okay, right. So this is the example if you have one single sample, right? And the outcome will be just one table with one abundance information and species identification. But what if you have multiple samples, right? You do the same process, the DNA extraction, the phase 1, the phase 2 sequence, and the biometric part you can combine them with data. And in one data, you can do a comparison between the sample A and sample B. So you'll be able to get the information. So what other species are highly abundant in one environment, which one is good in another environment. Okay? And the more sample you have with the replicates, then you can add up like other information like statistics, alpha, beta diversity, what else? We have differential abundance information and a lot of things that you can do with replicates. Okay. Right. So yeah. So next. Okay. So now I want to move into the demo report. So this is our result that if you subscribe to the service, this is the data that you will get. Okay, so feel free to scan code, the QR code. It will access you to our demo report, which you can download to your PC. All right, and you can open it and you can see how the result looks like. So you can see there's a lot of things you can play around. Okay, but this is a file you have to download. It's quite huge. You won't be able to open your phone. Okay, but I will demonstrate you how the result looks like from my PC. Briefly, I'll go through with the demo report because it's quite a lot of information here. Right, so this is our demo report. If you scan the QR code, so you will have a zip file downloaded with all these files inside. Okay, right. So once you have this, you can see there's a lot of folders. All these things are the data, the result that provide from the analysis. So a lot of things that you can get. You can get bar plot, heat map, pronoun, region, all these statistics analysis. Okay, but to help you to go through the result, you can just open the report. Right, so this is the report. Right, so this is the demo report for the input consequence analysis. I just want to show you just a quick part here. So this is an example of a total of our data set of 9 samples. 9 samples, and they are coming from 3 types of process. Okay, so there are process A, process B, process C. Each process they have 3 samples, 3 replicates, and in total have 9 samples. Okay, right. So we will compare between these 3 processes. So want to see what's the difference between 1 to 9. So yeah. So this is just assessment. Let's look into the main data. So this is the main data. Okay, right. So this is the sequence information, the sequence ID. Okay, and this column represents the abundance. How many of this sequence present in the that represent in each of the environment, each of the sample. So, for example, that you can see that this C sample, there are highly abundance of this ASP1. And within the sequence, when we match to the database, we know that this match to the database. Right, so we should match to the database. Interestingly, other sample doesn't have lactobacillus. So this is something that we can do. Right? And also, so you can see this is the relative number how many we have this first particular sample. Right, and using this basic data, we can convert to any type of information, like for example, the bar chart, the bar plot here. So using the bar plot at the genus level, so we can see the abundance, the profile of the microbes in the environment. So you can see that the C group are consistently highly present of the this is matched to the lactobacillus, 97%, right? 97%. So and the rest, what are they? So we know that they are matched to a small percentage of glucuronate, right? And what else? Acetobacter. So this is something that we can do to study a mixed microbe from the environment. Okay, so this is for C sample, A sample. You can see that Prevo data here. And then we have like for B sample, we have Acetobacter, coordination. So this is something that information that we can get. There are more things that we can get. For example, we can do a diversity study, the statistics. Right, so we can have like the alpha density and alpha density. Right, so to give information, how does this environment works? Are they rich of bacteria, low in bacteria? So the statistic can help you explain that. Right. And I also like to look into this data when I work on the which is the different environments. This data can tell you which microbe they are and which, like for example, using the next at genus level, right? And let's say we open this picture. So here we tell you that this is a group A, group B, group C. So in group A, highly present of immunosuppression. Group C, lactobacillus, something that you saw that's not 100%. The B will highly, we can see the Acetobacter, Acetobacter. So we can have this information, can do a comparison across all samples. Right. I will skip this part. You can continue. Use the QR code just now to download and have a look on the result. All right. And see what we get for this samples. So now let's continue back on this. Right. Okay. So that's about the amplicon sequence analysis. Next, let's move on to the shotgun metagenomics. Okay. All right. So shotgun metagenomics is actually quite very straightforward, easier than amplicon sequence analysis. Right. Same from the mixed microbiome community, but we have the soil sample. We do the DNA extraction. From the DNA extraction, we got all the DNA from this sample. We straight we go for library construction. Meaning we sequence everything, the DNA here. Right? The outcome here will be the raw data, the sequence data in FASTQ format. Okay, so this data will be very huge compared to the previous application that was analysis because we sequence everything. So normally we start with 6 GB of data. To compare how big 6 GB is that our human data genome is about 3 GB. So it goes to 2 times of our data, so it's quite huge. Okay. 6 GB is a minimum for one sample. So if let's say we have 10 samples, so we have 6 GB for each of the samples, total will be 6 GB. Right, so to process this data, so we need to do this. Okay, so that is about the sample submission, right? Sequencing, right? I mentioned just now, 6 GB per sample, run on the immunoglobulin set. That would be the most economical sequencing platform, right? And the outcome from this will be the FASTQ format in from this machine. Right? So we need to analyze the data in this stage here, which is the bioinformatics process. So since that sequencing here, the sequence is a very short sequence, so we need to reconstruct back the DNA. We have to assemble the sequence to make a longer assembly. Right? So this is the part where we do the co-assembly. Okay. So we have from the raw data in the FASTQ format. In the FASTQ format, we'll do the co-assembly. So we assemble all the data. Let's say we send 10 samples. So we take all the FASTQ for these 10 samples, and we assemble them all together. We call it as a co-assembly. So after the assembly, we have a quantic sequence longer than compared to the original raw data, which sufficient for you to do annotation to find the sequence, the genes inside this data. So from this assembled data, we can do the RNA ribosomal prediction, which we can use for taxonomic identification. We can do ORF calling, and we can do the annotation by matching to a database like CPI. So from there, you can find the enzyme, the KG, COG, class of orthodox genes, right? So we have this data table, and together with all this information, we can back to this, let's say, 10 sample. We can have the we can do a comparison analysis, right? You can do a comparison analysis on the taxa, what species, how many species present in the sample. Also, we can do a comparison across the enzyme, the functional information. How many genes are present in sample A? How many of genes are present in sample B? So this is something that we can do with the metagenomics process. Right, so this is some examples here, right? So the data. So again, we have the demo report for the short time analysis. Feel free to save this picture. You can download to your PC and you can look it later, right? So you can access what are information that you can get from this our demo report. So we basically some of the results. So you can see this is the taxonomy abundance at genus level. You can see what species present in the sample, how many are there in the abundance. You can also have the functional information abundance, KG, COG. You can also have the what you call the statistics analysis as well. So just a short one. We'll go to the demo report. Right, again, so this is the for short-term metagenomics. So you can see we have the result, we have run analysis, and we get all the results in the respective folder. To make things easier for our customer, you can just access the report.html again. Right, make it slightly. Okay, right. So you can see this is result for short-term metagenomics. Let's look at the statistics. So this is example for 6 samples. So we have 2 groups, group A and group B, to environment. So each environment we have 2 replicates, right? So replicate A, so we have group A, group B, each have 3 3 samples in each group. Right. Yeah. So let's look into the taxonomy. Right. So yeah. So this is the result at the genus level. So if you want to see at other level, you will also have access by looking at the level. This is something that what you can see in the sample. So for example, in the group B, we can see this is of the microbes, which is matched to the something matched to insect. So this is an insect actually. So this sample are highly advanced of insect. A group is a mixed sample. You can see it's matched to the phylum of bacteria, gamma protein bacteria. Okay, bacteria. So this is conifers. Okay, so we can see the mixture of organisms in the sample. The good thing about the sugar metagenomics, because you don't need to PCR the sample, so you don't have to like specify do a target specific capture. So let's say bacteria or fungi. For sugar metagenomics, you don't do that because sugar will sequence everything, meaning that one sample you can get information across all organisms. So for example, you can see there's a bacteria, you can catch bacteria. So bacteria group, we have it's a long list actually. There's a bacteria, so there's a long list we have from the Excel file actually. So we have the Excel file to present, but we match to white group, so we can capture fungi, bacteria, up to even virus. So right. So that's the advantage of shotgun methodology. So it's not a group specific analysis. For amplicons, you have to define. If you want to capture bacteria, you have to use CPS. If you want to go for fungi, you have to go for IPS. So very you need to focus on specific group for right. Right. Other than that, it's more or less the same. So you can have you have a KGG. So for example, you have like this A, B, and C. So A and B. So this is the enzyme ID. So you'll be able to see that what are the enzymes highly abundant across. Okay. So like for example, you can see the alcohol, the hydrogens are highly abundant in the A group, but low in the B group. So this is something that you can do. Direct comparison. Other than that, we have alpha diversity, statistic analysis, and so on. Okay. So I will skip this because there's a lot of information here. You can go to our demo report, and if you have any question about this service, this result, you can contact us. We can have a session with you to go through with the information that contains in the demo report. Right. Okay. So we will set this picture. Okay. Right. Okay. Yeah. Okay. So we almost come to the end point here. The last part. So the last part will be the things that will assist our customer when they do the amplicon sequence analysis for sugar metagenomics. Normally, when people do, when the customer they go for this service, the metagenomics, they don't send one sample. We have a customer that send up to 1,000 samples. Right. So when they have 1,000 samples, the challenge is that they might need to rearrange the data. So I might want to show the first phase of result, the second phase, or maybe I need to show the first 100 and then second 100 in the second paper, right? So initially, normally the data will come in like the one file. Everything will put inside this 1,000 sample in one file, right? But using this our cloud base, the customer can upload the data to this cloud and they can refine back the result. So you can refine back according to what they want. I want to show the first phase of my analysis, the first time point, maybe first hours or maybe first 5 days. So they can rearrange back the data using our cloud. Okay. So for example, just now, let's say you can see the result. Or maybe the show you the demo report just now, right? So for example, the demo report. here, this is the demo report for every cloud, sorry, for the every sequence analysis. Right, so let's say now we have A, B, and C. Using the our cloud group, we want to show A, B, but exclude the C. So you can also use it our you can analyze back data. So meaning that you can define back so what you want to present for your publication, for your report, and so on. So this is what our web tool can do. Okay, so this is very useful if you have a lot of sample, right? And this service came to our customer for free, right? So anyone who will subscribe to the service through Nam Anh first base and use our standard package, they will get this access to the cloud for free. Okay, yeah. So they just a web-based tool. You can just take a picture and then there's the code that you can access, and that's what they call the trial kind of like data set. You can upload the data set and play with the features in the account. Okay. If this code only lasts for 30 days, but if you want to expand it, you want to play around further, you can let them and you can give it another code. Right? Okay, right. Okay, so that's last of my presentation. So yeah, so now I open to the floor if there are any questions related to this metagenomics, the consequence analysis or sugar metagenomics, or anything about NGS. We'll be able to. I will try my best to assist. Any question? Or you not clear anything, you can also let me know. I can try to.
I have a question. So like the presentation, so in the presentation we have two kinds of tasks, right? And
yeah.
One is the sequence and another.
Yeah.
So in the first one, after you harvest the sample in our lab, so what did you do? We just extract the sample and then send direct to the company, and the company will perform PCR and other steps, right?
Yes, correct. So you can send the DNA sample to Nam Anh. Nam Anh will ship to us, and we do the sequencing in our lab. So you don't need to do the PCR step.
So okay, in terms of price, I also mentioned the Andikan is cheap, right? How
yeah.
cheap?
For the price, I let Nam Anh talk to you. Maybe he get some special price for you, so I let him talk to you on the price.
Because we also, I also wonder if it's so expensive. How compared with the Sanger of one?
For Sanger, I would say that it's definitely Sanger is very cheap, but it's only target one sample, one bacteria. But if you're looking for something that mix Illumina, would be kind of like a bit expensive. But relatively, if you calculate the amount, the cost per base, like how many you get for base, actually Illumina, this NGS is very cheap. In terms of perspective, for example, we sequence a bacteria on Sanger, right? It can cost you like in USD could be up to like using Sanger alone. Sanger it could be up to 10K USD. But nowadays for Illumina alone, you can, because its cost per base is very low, can go up to like 500 USD or maybe around that, around 500, 700 USD. It could be like this, but not as expensive as Sanger. Sanger is good if you are doing a small part like plasmid sequencing, short sequencing. It's good for Sanger. It's cheap, one sample. But for large data, NGS technically relatively is quite cheap. So yeah, per sample, if you get the number of sample, yeah, it might be more than Sanger. But in terms of information that you can get, I would say it's quite cheap actually. But the pricing, the amount of data we can talk to Mr. Dan.
I have another question relating to the process for bioinformatics. So after we submit to just a company, what we can get from the company?
Okay, good question. So we will once we process, we sequence the analysis we provide the information, the table, that's the result, and we also provide you with the raw data, the data from the FASTQ file. So you get two set of things. So the raw data and also the analyzed data. The analyzed data will be shown in the folder just now. So the one that the QR code just now, that's the final result.
So this way we have the raw data, so we need to do our analysis.
You don't have to. You can use data is already processed for you. You can just open the result for you to see what's the content inside data. But we do also have a customer that requests only the raw data. So meaning that after the sequencing, we just give you the raw data without the analysis. So the service will be separate. So some customer want raw data only without analysis, but most of the customer will have one both. I want the raw data and analysis together. So it depends on what you want for your project. So we can deliver both.
Yeah, I have a question. Thank you for your presentation, and we really interested in this the metagenomics application, and we'd like to ask about the data set. When we upload the data set to the your website, and how to be because we worry about the copyright of our project, and and how we have the like acknowledge between us, and
we understand about the copyright concern about the data might be linked to other parties. Okay, so normally, not normally, this is our standard analytic process. Once we complete the sequencing and analysis, we'll release the result to you, and we'll let the customer know that please get this data within 30 days. Please download to your PC the data, the sequence, and the result. After the 30 days, then we'll start to push the data from our service. Okay, because we do a lot of sequencing, so we don't have big server to keep all the customer data. It will be constantly replaced, constantly delete. So you don't need to worry about your data once downloaded. We only keep like for another few months just in case that you might want to have the data again. So you need to contact us as soon as possible before the data will be deleted. So our policy, we won't keep our customer data for long. So after the process you have downloaded, we will delete it. So the most will kept by request unless the customer requested to have longer time than but no, we won't keep for long to ensure that this data won't leak to other parties. Okay. We will really state that in our email. Please download the data before we download it. We delete it from our server. So that's our commitment to keep the privacy of the customer data.
Yes. And I have one more question about the data, and how we work with our computer, because I think the shotgun metagenomics is very big data, and very hard to work in one maybe laptop like this. And how we analyze and do all the things between shotgun metagenomics and American.
That's a very nice question. But we understand that most of our customer doesn't have big server, super computer. So the result that we have is only processed and analyzed data. So it should be good. The processed data can be opened on PC because it's only like a table, Excel, just HTML format. It's very small and can be navigated in a small PC. As long as you have like nowadays you have like 4 GB, 8 GB PC laptop, right? It should be good for you to navigate around. The problem is if you want to start from the raw data, that will take a lot of power. Super. You need to have like a server to analyze data. But if you subscribe to our analysis service, we have done that for you. So you don't have to run the process because we have done it for you. So what you need to do, you just open the table. Even the result, if you can see the result for the shotgun metagenomics. So you can see all these things are processed. Okay, like for example, the taxon.
So that is the whole one, right? The whole service for the result.
Yeah, this is just for the result.
Yes.
Okay. And you don't need to analyze anymore because we have summarized, we have put in Excel file. So you just need to open the Excel. So you can see like the abundance, everything with Excel. So it's a small file.
Mm-hmm.
So this is the file, process file. So you will get everything in a single format. So
Yes.
you don't need to have a supercomputer to analyze the data. So you just have to work with Excel, small file, table, okay.
Yeah.
So you don't need to purchase any supercomputer to analyze this data. Okay, and in case that you have this big data and then you want to, let's say, I want to refine back. Just want to show, remove the A3 because the A3 doesn't show good data. Want to show A1, A2, B1, B2, so you can use our cloud to refine. Meaning that you don't need to use any computer to analyze data. You can use our cloud to refine back the result.
Same price.
The amplifier is free.
Oh, really?
So we give you for free.
Yes.
So you can, but you have to subscribe to the service first. Then we give the link for free. So let's say want to change, then you can change the data. You access to the Apple Cloud, and then you can adjust the data.
Okay, thank you.
I'm wondering the result will just as the genus level. How about if we interested in the species?
I like this question. Okay, right, because this is only a kind of very short presentation. I don't go in that deep. Okay, this question I want to for shotgun metagenomics, it doesn't we can capture up to species level. That's for shotgun. Depends if because we have what we call the buffer. It depends. Most of them will be captured to species. That question related to amplitude sequence analysis. Can we come back to this slide?
So we just apply for the genus and
there's two things that I can tell you. Okay, right. The gene marker I want to show you. Okay, right. It's your. I like when you pick up this point here. Okay. Okay, right. This thing. When I talk about the what they call the amplitude sequence analysis, we normally we sequence using the Illumina platform. Okay, the limitation with the Illumina platform is that it's considered as a short sequencing platform. So the longest we can go is about 550 base pair, right? Bacterial CPS ribosoma RNA, the full length is actually about 1.6 kg. Okay? But Illumina cannot capture the whole 1.6, 1.5 kg. So what they do is they will capture part of the variable region. So for example, mostly we do the video. If you go for publication, they will go for capturing V3 and V4. Right? You have the two variable region. Sufficient, they have a variable, high variable on this region, sufficient enough to do the taxonomy identification. We need this variable region to see the to kind of like identify. Oh, when you have this signature, this kind of like sequence, base sequence, we can match to this species. This different, we match to another species, right? But this V3, V4 kind of base pair only good enough up to genus level. You don't have enough resolution to go to species. You might have species information, but very limited. Okay, right. So that's for Illumina. Okay, if your analysis sufficient to go for genus level, you can go for Illumina platform. But if your objective, if your project, you need to have up to species level, then we can suggest you to go for another sequencing platform, which is the third generation platform, which is the ONT number 4 or PacBio. We offer number 4 service, right? So since number 4 can sequence the longer reads, so instead of with PCR V3 V4, using number 4 we can cover from V1 to V9. So we have a V1 to V9, and from this information we can capture species information. Okay, we are trying to see the demo report for the full name. Is that pick up this question? Yeah, we have number 4 here. Right, so this is the result for full name. This result for full name. Right, and you can see from the taxonomy overview. So you can see that so this is upper Aspergillus, is genus, Aspergillus fishery is species. So you need to use a different platform to capture up to species level. Right. So and since that is using a third generation platform, the price is slightly expensive compared to Limina. Right. So we have that solution. So it depends what's your objective. Then if you need species, then I'll go for full length. This is good if you're working with, for example, studying infectious, let's say like the tomato, you want to know what's the infection because genus is not enough. My project study working in Bucoliria, Bucoliria is a genus. Bucoliria pseudomale is a pathogen to human, but Bucoliria epidermidis is good for plant. So if you just know Bucoliria alone, it's not enough. So it depends on what's the objective. If the species information is good, is important, then have to go for full length. Any more question? About from the floor at the back? Or maybe I can ask you a question? Right, so no more question, right? So maybe we can move on to the next one. Okay, the next one will be the next one will be the session. It's time for me to ask you. Right? So we have gone through the presentation. Okay, right. For the quiz session, we have some prizes. So we have like how many left? 5, 6. How many left? I think we have 7. So we got So we got 7 prize, 2 ice box here, so we can use this to track, keep your sample, and then go to places. And we have 5 flashes, like this, like flashes. So we have 5. So we're looking for 7 winners here. Okay, right. So ready? Okay, first question. What is the difference between the amplified sequence analysis and short-term metagenomics? Các bạn ơi, dễ lắm. Tiếng Việt cũng được ở đây. Đấy về cho khoa kìa. That's a paper that I showed you.
One is need PCR and need PCR.
Có anh Sơn sẽ hỗ trợ với em. How about shotgun? Shotgun?
Shotgun you don't need PCR.
Right. So for Amplicon you need to PCR. For shotgun you don't need to PCR. Which one you want? Either Bashir or
skip for the faculty.
It's okay. You have a few more. Which one? Choose. You can choose. I don't know.
Maybe student.
Oh, okay. You can choose. It's okay. You can pass to.
Yes. Okay. Thank you. Yeah.
Okay. All right. So you got one answer. How about we look for another two? Yeah. Yes.
Information application.
Yeah. You.
So that functional information. But
Yeah, correct. So amplified sequence analysis doesn't have function information because you only capture the DNA marker, no gene information. That's why you will have a function information. For shotguns, sequence editing, you will get the text information as well as function information. Alright. So which one you want? The flashes? Oh, you can collect. Come on, select. I know this. Okay. Okay. Yeah, we can have another one more. One more. It's very simple. It's related to the gun. You need to consider before you go for Applicon. Can you afford it or not? Right? Yeah, I'll give you some hint. If you're shy, you miss the opportunity to get it. It's very limited edition. Right? You want to try? Don't be shy. Otherwise, you cannot end the session. Right? Something related to the budget. Okay. Right? You need to see whether you can afford or not. So if you are rich, your project have money, then you can go for another one.
And cheap one is expensive.
Yeah. Yeah, because somebody has to give some answer. I want to try. Want to try?
Ở đây.
Let's get in the subject. Correct. Okay, right. So we have the third winner here. So yeah. Okay, correct. So let's move on to the next one. Okay, right. I mentioned this type. So name of the DNA markers are used for because analysis and its target organism. Right. So just now there's a picture showing that there's a few. And what's the target the DNA marker?
Đó là tiếng Việt thôi.
How about Prof? You want to ask? Okay, maybe I can do something. All right, so for bacteria, what gene marker we can use? Anyone?
CPS.
For bacteria, want to try? We have somebody can help you. No worries.
Buổi sau ấy, buổi 16 ấy.
Maybe you can give her one more time. Right.
Okay.
Vegetarian sickness.
CTS.
Alright. Right. Okay, so for bacteria we use CTS. Okay, how about for fungi? Fungi?
Yes.
You can a lot of things. Alright, for fungi, what is the DNA marker that we can use to target? Ishai, this is a special limited edition. Want to try anyone? Maybe I can point out, like, how about you? Yes. How about you? Want to try? Maybe we have friends who oh, give it a try. Don't be shy. So you need to have one track. Okay. Yeah, okay, yeah, fine. Okay, for fungi, use ITS. Alright, so another one. So maybe your friend, next one, want to try? So, okay, how about for animal? For animal or other?
Mấy bạn nhiệt tình tham gia đi còn quà nha.
The export for the kid. It's the export. Oh. Oh, okay.
Yeah.
I
Nhiệt tình trả lời đi.
Oh, okay. Right. Okay. Right. Okay, last one. Last one. Yeah. So, okay. Want to answer? Okay, good. Yeah, that's the correct answer for that animal. For the animal. Okay, last question. Right. So last question. What is the web tool name? Just now we talked about the web tool name. What is the web tool that you can find in the okay, something. So the name is somewhere around there. What's the answer? Need to get somebody encouraged to. Come, we have like last one. Anyone to answer the question? There's some hint now. Right? There's some maybe re-accession, not doing. I don't know which one. So we can try out. Yeah. Yes. No, yeah, it's amplifier. Yeah. Thank you very much. Okay, so that's the last of the session. Okay, so we have ended the session today, right? So if you have any question regarding the NGS or today's topic about metagenomics, you can feel free to contact Nam Anh. Right, they will be around to assist you. Okay, and just for today's join the seminar today, we have some special exclusive promotion for our those who attend today. Okay, feel free to catch Mr. Tan before we run away. Okay,